Research in Chinese medicines is ongoing, from the identification, efficacy, to safety, accumulating valuable knowledge and experience. Since the handover, Hong Kong has worked to promote the scientific research and modernization of Chinese medicines, achieving substantial results to date.
Right. This is the first step in making medicinal oil. This extraction barrel is what we call an oil still. These are qualified Chinese medicines. Here is the lacquer, already measured out, so we can just put it all in. It's heated for 3 days to extract its essence and dissolving into the oil. After extraction, it's pumped there to sediment for 14 days. Each barrel has a status label, showing when it's cleaned, when oil is added, and when it's pumped out. There's also a sampling point on this barrel. The extract must pass inspection before being pumped to the next stage of the process.
Mr. Kwok's pharmaceutical factory has been certified with GMP since the 1990s. He's one of the pioneering local manufacturers of proprietary Chinese medicines, employing modern production and management methods. GMP stands for Good Manufacturing Practice, a quality assurance system widely adopted around the world.
This is a clean room. The floor is coated in epoxy. The corners and the base of the walls are all curved to facilitate cleaning. This high-efficiency particulate air filter removes 99.99% of suspended particulates.
The temperature, humidity, and pressure are all controlled.
This room is dedicated to the final step of the production process, which is mixing.
Local manufacturers of proprietary Chinese medicines' transition towards GMP standards represents a significant milestone in the industry's journey towards modernization.
Before moving to the Wangchukang factory, our old facility was in Causeway Bay. There was a room for the oil still. Secrecy was highly valued. Acting on my grandfather's instructions, the pharmacist would measure out ingredients inside that room. When the final product was out, we relied on its smell and feel when applied. Unlike these days, there were no formal methods for testing. It was really about manufacturing with integrity. We obtained Australia's GMP certification in 1998, and then Hong Kong's GMP certification in 2013. While we wanted to continue improving, mainland China began requiring Hong Kong's proprietary Chinese medicines to be GMP certified for sales there. The factory is my grandfather's century-long legacy, and we hope to do even better and pass it down.
GMP certification for proprietary Chinese medicines in Hong Kong is not mandatory. However, to strengthen the regulation of these medicines, since 2004, all proprietary Chinese medicines manufactured, sold, or exported in Hong Kong must be registered with the Department of Health. Mr. Li Yingsang, the first chairman of the Chinese Medicines Committee, was responsible for promoting registration of these local products. He is well acquainted with the challenges faced by the industry.
There were about 20 to 30,000 proprietary Chinese medicines products on the market, which we had to regulate within a very short period. It wasn't about just assigning them numbers. We needed various proofs, such as interpretation of formulating the prescriptions, ingredients, methods of administration, and some test reports. One of the most time-consuming and expensive aspects in the course of registration was on the development of certain methods to test their various components, which might not be 100% successful. If a method couldn't be developed, hundreds of thousands or even millions of dollars spent on development could be gone for nothing.
To assist the Chinese medicines industry with testing and quality control, the Department of Health's Government Chinese Medicines Testing Institute actively collaborates with the industry. They develop various testing methods for Chinese medicines for use by the industry. The testing method for medicinal oils was the first research project undertaken by their chemistry laboratory.
First, we needed to understand the chemical markers present in the medicinal oils available on the market. What we found is that most medicinal oils produced in Hong Kong contain methyl salicylate, camphor, and similar ingredients as their primary chemical markers. And based on these particular chemical markers in the oils, we worked to develop relevant chemical testing methods that could be used across the entire industry. In fact, the production of proprietary Chinese medicines require using various excipients, so the interference from the matrix is very severe. Take medicinal oils as an example. Excipients include palm oil, lavender oil, and also clove leaf oil. The related analysis is challenging. Fine-tuning the experimental conditions is the most time-consuming part. Adjusting variables such as temperatures, ramp rates, or hold times to separate the related matrix interference, ensuring our target chemical markers are unaffected by any interferences. So this spike on the screen here is pinene in turpentine oil, and this one here is eucalyptol, which we separated.
The Government Chinese Medicines Testing Institute was established in 2017. Developing analytical methods for the chemical markers in medicinal oils is just one of their many research projects. For Chinese medicines and decoction pieces, the Government Chinese Medicines Testing Institute has conducted extensive research on origin tracing and authentication.
The GCMTI has published 10 volumes of the Hong Kong Chinese Materia Medica Standards, which we refer to as HKCMMS. These volumes cover 330 commonly used Chinese Materia Medica in Hong Kong. The volumes also include various identification standards and methodologies. We also published the Compendium on Identification of Easily Confused Species of Chinese Materia Medica in Hong Kong by macroscopic and microscopic characteristics, employing macroscopic, micromorphological, and microscopic identification methods.
Macroscopic identification, which involves observing, touching, smelling, and tasting to differentiate Chinese medicine, is the most convenient and commonly used method. Take cordyceps, a commonly used Chinese medicines in Hong Kong, for example. It looks alike with its easily confused species, Cordyceps hawkesii. Researchers at the GCMTI have compiled several key points for differentiation to help the industry and the public distinguish between them.
We can examine their stromata, which is the black part at the top. So typically, cordyceps have only one stroma emerging from this point here. You can see the top here doesn't have significant swelling. However, in some cases of Cordyceps hawkesii, where they have just one stroma and no significant swelling at the top of the specimen, that's the time when we will need to utilize other methods to assist us in differentiation. One of the micromorphological distinguishing features is the spiracle. This one here is the spiracle of Cordyceps, and this is the spiracle of Cordyceps hawkesii. Upon measuring the spiracles, we notice that the Cordyceps hawkesii spiracle is noticeably longer than that of Cordyceps. The project of identification of easily confused species of Chinese Materia Medica in Hong Kong by macroscopic and microscopic characteristics studied 100 pairs of Chinese medicines. We didn't just list their basic physical characteristics, but chose the most critical and easily observable features as key features enabling quick differentiation.
For Chinese medicines that cannot be easily distinguished by morphology, microscopic identification is one of the effective methods. Researchers summarize those features of the Chinese medicines that are clearly observable under the microscope. This will aid the industry to accurately identify Chinese medicines.
This is Cortex paraplaki and Cortex acanthopanacis, which look quite similar in appearance. We can examine the features of their powder. For example, Cortex acanthopanacis can be seen with clusters of calcium oxalate with many sharp angles resembling crystal clusters. When observed under a polarized microscope, they shine. This is the powder of Cortex paraplaki. The crystals are clearly different from those just seen. They are square-shaped.
We consulted industry representatives and members of the advisory committee. We wanted to know which type of research would be the most worthwhile and beneficial for Hong Kong. They mentioned Pilo's antler as a focus.
Here are some Pilo's antlers. This one is from Jilin. This one here comes from New Zealand. And this one is from Canada. There are also some very old ones on the table that have become calcified. It's easy to differentiate them like this. But once they're sliced, it becomes difficult to tell from which country they come and their different species.
The challenging thing about the different deer species is that they cannot be distinguished based on chemical markers, and we have to employ DNA technology. We've studied the DNA of deer species like Cervus nippon temminck and Cervus elaphus lineus. Using simple nucleic acid techniques, we can now differentiate authentic products from adulterated ones. At the Government Chinese Medicines Testing Institute, our primary goal is to enhance the quality, safety, and testing methods of Chinese medicines to gain international recognition. The ultimate aim is to elevate the international profile of Hong Kong's local Chinese medicines industry, positioning Hong Kong as an international center for Chinese medicines testing. These exhibits are basically items that the Customs and Excise Department handed over to us in the 60s and 70s for verification of whether they were prohibited items. The counterfeits are sometimes made to look more authentic than the real ones. Here we have fake birds' nests and fake cordyceps.
Among the tertiary institutions in Hong Kong, the Chinese University of Hong Kong stands out as a pioneer in Chinese medicines research. Since the 1970s, CUHK has conducted research in areas like identification and pharmacological studies on Chinese medicines. After the handover, they have further consolidated interdisciplinary researchers to enhance their research capabilities. Professor Leung's team has been working on a Chinese medicines research project for over 20 years, utilizing a compound formula of danshen root and kudzivine root to help patients with cardiovascular issues improve their conditions.
In modern heart-related Chinese medicines, danshen root is always present, though kudzivine root may not be. The proportion of danshen root to kudzivine root requires careful study.
Universities applied modern method on research and development of Chinese medicines. Steps from extraction to various efficacy and safety experiments are all rigorously performed. The starting Chinese medicines materials must first be identified and tested in the laboratory to ensure they meet experimental standards before extraction begins.
In this bottle here, I have our result. This is the extract we obtained after concentrating and freeze-drying the danshen root and kudzivine root.
The powder obtained after drying serves as the foundation for various subsequent experiments. In verifying the efficacy of the medicines, the research team conducts tests following protocols in modern drug development. Cell experiments are one of the frequently used techniques in this process.
If the vascular smooth muscle cells are in pathological state, they'll proliferate and migrate to the inner walls of the vessels, leading to arterial blockages. This condition is known as atherosclerosis. So what we can do is create a cell culture environment and add our traditional Chinese medicines into the culture solution. We hope that danshen root and kudzivine root can inhibit the proliferation of smooth muscle cells and reduce their migration to the inner walls of the blood vessels, thereby improving the condition of the blockage. We then performed a step called staining that assisted us when observing the smooth muscle cell colonies, which appeared as small dots. The more dots, the more proliferation. With the addition of danshen root and kudzivine root, the number of dots decreases, clearly indicating their effects in inhibiting the activity of smooth muscle cells. One of the most common cardiovascular disorders is hypertension. This experiment aims to test whether danshen root and kudzivine root can be used to induce blood vessel dilation. We obtained the aorta from a mouse. Then we cut it into rings and hooked it onto a device. The computer shows changes in its force, indicating whether the blood vessel is relaxed or the vessel is contracted. We are now adding a vasoconstrictor to the experiment to simulate high blood pressure condition. As soon as we add that, the force quickly rises to 2.2 grams. This rise in force indicates vessel contraction. At this point, we will add the extract of Danshan root and Kudzavin root. And the value decreases. We will accumulate the concentration continuously to see if increasing the concentration will further enhance the blood vessel dilation.
Results from laboratory research may not always translate directly to human bodies. The research team eventually collaborates with doctors from various specialties for clinical trials of the formula. Professor Leung's Danshan root and Kudzavin root formula has already undergone its 5th clinical trial. Since blood vessels are connected to the entire body and many diseases are related to them, there's still a vast scope for research on the therapeutic effects of Danshan root and Kudzavin root. At the University of Hong Kong, the director of the School of Chinese Medicine, Professor Feng Yibin, has been researching golden thread, an extremely bitter Chinese medicine. Does the saying, "good medicine tastes bitter," have a scientific basis?
Golden thread is in China's first herbal compendium, the Shennong Ben Chao Jing, classified as a top-grade herb. Over 1,400 formulae containing golden thread were recorded in the Song dynasty. Over the past 20 years, my focus has been on studying the golden thread's monomers, uses on its own or in combined formula. I have studied the application of golden thread in diseases such as liver disorders, tumors, and metabolic diseases.
Professor Feng's research team has tested golden thread's therapeutic effects on several types of cancer. They have also observed positive results in reducing the side effects of chemotherapy and in treating eye complications caused by diabetes.
Currently, we are conducting an in vitro cell experiment to investigate golden thread's effects on tumor cells. This is a culture flask with cancer cells. A researcher is adding the golden thread extract into the cancer cells. And then we'll observe whether golden thread can kill the tumor cells. We can see that some tumor cells have shrunk, indicating that golden thread does in fact possess some degree of anti-tumor activity.
Beyond cell experiments, the research team also uses animals, such as mice, to study the anti-cancer effects of golden thread.
We inject the cultured cancer cells into the corresponding organs of animals in order to create relevant cancer models for our research. We then administer golden thread to these models. From there, we begin to monitor the tumor growth that is occurring in the animal bodies, which enables us to evaluate whether or not golden thread has any anti-tumor effects. We're also hoping to understand how it works. We perform network pharmacology analysis on each chemical component extracted from golden thread to study their biological targets.
To gain global acceptance of Chinese medicine, its scientification, modernization, and internationalization are crucial. In other words, we will need to apply modern scientific techniques to reinterpret the ancient languages and explanations and applications so as to elucidate its mechanisms of action and biological targets for people to understand today. This is our primary task.
In 2022, the National Administration of Traditional Chinese Medicine invited the Department of Health to participate in the National Survey of Chinese Materia Medica Resources. This is the first time Hong Kong has ever been involved in this survey. On this day, Professor Zhao is leading researchers in preparation for the survey.
There's quite a lot of grass leaf sweet flag in this area. Over there, we can see birdwood's mucuna.
Right here?
See? This is a plant from the Fabaceae family, known for its use in relaxing tendons and unblocking meridians. It was first discovered in Hong Kong over 100 years ago. That makes it a representative species of Hong Kong.
This part right here is its calyx. With 1, 2, 3, 4. See that? It has 4 lobes. The color of the flower, when it's fresh like this, it's yellowish white with a hint of green, or light green. Now, if it dries, it turns black. So let's take note of this characteristic. Let's also record the GPS location.
The Hong Kong Department of Health places great emphasis on the National Survey of Chinese Medica Resources. They promptly organized a team and, over the course of half a year, compiled a preliminary list of Hong Kong Chinese Medica Resources.
Oh, I see one.
With
There's
flower.
one here. See it there? See it?
With flower. It has a
Yeah.
flower, but it's not very red. Here we have a Dunstar anise.
Which typically grows in streamside habitats.
Our aim is to ascertain the location and quantity of these plants. Additionally, as medicinal plants, it's important to understand their effects and uses. Information not recorded in ancient books but known through folk experience will need to be gathered and investigated through this resource survey. This will allow more people to recognize and apply this knowledge.
The survey of Chinese Medica Resources is just one of the many collaborative projects between Hong Kong and mainland China. In recent years, Hong Kong has continually strengthened exchanges with the mainland and the world, fostering the transmission and innovation of Chinese medicines.
Hong Kong has always been benefited from the solid foundation of Chinese medicine in our motherland. We have signed many agreements with the National Administration of Traditional Chinese Medicine and the National Medical Products Administration, engaging in deep exchanges on regulatory aspects, drug inspections, and Chinese medicine policy. Additionally, under the framework of the Guangdong, Hong Kong, Macau Greater Bay Area, we have signed several agreements. The Department of Health has been actively involved in the internationalization of Chinese medicine, working collaboratively with the state in various platforms to promote international standards in this field. Looking forward, the Special Administrative Region government will continue to maintain close ties with stakeholders in mainland and the world. Under the principles of mutual benefit and complementary advantages, we hope to deepen exchanges and cooperation in the field of Chinese medicine with various regions, working together to build a healthier Hong Kong and further integrate into the national Healthy China initiative.